So thank you, Peter, for the nice introduction. So hello, everyone, and today I'm going to present my talk, which title, wow.
I think you have to hit yeah.
Yeah, I understand. Okay. Okay, here we are. Okay, so today I'm going to present my talk titled Deciphering the Driver Mutation Puzzle in Cancer. So DNA molecules in human cells are constantly exposed to various mutagenic processes, resulting in accumulation of alteration over the cell's lifetime. These mutational processes can arise from both endogenous and exogenous factors, including DNA damaging agents, error in DNA replication machinery, and deficiency in DNA repair mechanisms. And in terms of alterations which are common in cancer, there are three major groups. Small mutations, which include single nucleotide variants, insertions in deletions, chromosome rearrangement, which include copy number variants, and they affect the overall organization of chromosomes, and copy number variants, which include deletion, duplication, and loss of function, loss of heterozygosity. So, and the vast majority of mutations which are common in cancer, they represent single nucleotide substitution, which will be a topic of my talk today. So, somatic mutation in cancer cells are identified through the following process. So, when an individual is diagnosed with a cancer, the tumor sample is obtained and the whole exome or whole genome sequencing is performed. In a parallel, also the normal sample is obtained from the same patient and also performed whole genome or whole exome sequencing. So, and by comparing these tumor samples with non-tumor samples, somatic mutation specific to this patient can be identified. So nowadays, thousands of tumors genomes have been sequenced, and there are many publicly available data sets of tumor mutations provided by such projects as TCGA, the Cancer Genome Atlas, CGAs, the International Genome Consortium, and many others. So tumorigenesis is a multiple stage process. Starting from early development, cells undergo multiple rounds of cell divisions and are exposed, as I mentioned, various mutational processes which generate mutations. And most of these mutations are supposedly neutral and they are called passenger mutations. However, a few of them drive cancer progression and they are called driver mutations. But it's crucial to highlight that classification into driver and passenger mutations, it's not binary. There are some mutations which are called latent driver mutations, and they can become drivers at particular stage of cancer evolution or when combined with other mutations. Also, individually infrequent and functionally weak mutation may also collectively become drivers. So the number of driver mutation may differ between cancer types and patients, and it's important to say that there are hundreds of mutations with unknown driver status, and there is no direct way to assess tumorigenic effect of mutations. So why is it important to identify driver mutations? So first of all, it's important for understanding molecular mechanism, molecular mechanism of cancer development, and as a consequence for developing diagnostic, prognostic, and treatment strategies. So there is a relationship between the presence of driver mutation in a cancer genome and the clinical phenotype of a given patient. So in some cancer types, patients harboring driver mutations might have better worse survival compared to patients without driver mutations. So let's explore current examples of driver mutations serving as cancer biomarkers. So, and the first example of this is driver mutation lysine to methionine in histone H3. So children diagnosed with diffuse glioma carrying these mutations in histone H3 exhibit the overall survival rate less compared to survival for patients with this wild type protein. So the second example is patients harboring specific driver mutation in the POL-E gene, which encodes DNA polymerase epsilon. They have better survival compared to patients without driver mutations. So in addition to individual mutations, clustered mutation in CP53 and BRAF genes have been recently found to be associated with overall survival. So in other words, driver mutation identification can be used in personalized cancer medicine. So a tumor sample of a patient can be sequenced and to obtain genomic alterations and identify driver mutations. And armed with this knowledge of specific driver mutations, it's possible to make informed decision about the most effective treatment strategies for these patients. So, and the overall goal of my research is to develop novel computational approaches to enable identification of driver mutation in cancer. So, and I'd like to start with the question, how to find driver mutation in cancer. And currently there are three major groups of methods: functional impact of mutation methods, recurrent-based methods, and probabilistic and machine learning methods. So, and I'll begin with by exploring the first group of methods which focus on the functional impact of mutations. So the main idea of this method is that mutation affecting functionally important region in proteins are more likely to be drivers. So if mutation has impact of phenotypes, in other words, it's more likely to be driver. So, and we know that mutation might have different effects on proteins. So they might affect protein stability, protein DNA binding affinity, protein binding affinity, and they can lead to conformational changes. So they can be loss of function mutation, gain of function mutation. So, and driver mutation have a higher impact on function, so high impact score compared to other mutations. So these red bars indicate the driver mutations. So next group of methods is recurrence-based methods. So today, recurrence of a mutation is remains one of the most reliable markers of mutation driver status. And let's consider two examples for better understanding. So we have case one: mutation A is observed in 10 patients out of 150 sequence patients. So this is our cohort one. So case two, mutation A is observed in 10 patients out of 15,000 sequence patients. This is our cohort two. In which cohort of samples, in which cohort of patients we are more likely to find driver mutations? So let's relate it to this simple example of coin toss. In coin toss, we have two outcomes, heads and tails. So, and we can use the binomial distribution to estimate the probability of getting, as an example, of heads. And this probability, depends on the probability of success in each individual trial and the total number of trials. So it has the following parameters: K represent the number of heads or number of successes, N is the number of trials, and P the probability of success on each trial. So let's keep all calculation for our two cohorts, and let's assume that we calculated two probabilities for cohort one and probability of getting driver mutation in cohort two. And so the larger number of trials increases the overall likelihood of observing 10 mutations, these recurrent mutations, by random chance. So in other words, if we talk about observing recurrent mutations, then it's more likely to observe such mutations by random in a larger cohort size, where cohort size is larger. So, and moving forward, we can see that this methods, this recurrence methods, depends on the sample size, the cohort size. So, but there is another factor that should be taken into account. This is background mutation rate. And this is background mutation rate, it's a rate. So basically it represents the baseline frequency for mutations that happen naturally during such processes as DNA replication and DNA repair. So, and somatic mutation rate exhibits significant variation across different regions of the genome occurring at various scale levels. So variation in mutation rate can be observed at large scale, it can be observed at small scales, and it also can be observed at mesoscale, bridging these two extremes. So let's explore mutation rate variation at the large or megabase scale. So it has been shown that the cell type specific epigenetic and genomic features, such as replication timing, expression level, and chromosome accessibility, might explain up to 86% of variance in cancer genome at this megabase scale. So, and one of the first features to be associated with mutation rate at this scale is gene expression level. So highly expressed genes tend to exhibit a lower mutation rate than lowly expressed genes. Also, an association between replication timing and mutation rate and replication timing has been also shown. So the mutation rate of later replicating genes is significantly higher than the replication rate for earlier replicating genes. So another large-scale feature is chromatin states. So we know that the nucleus is organized into two compartments, two large-scale compartments known as compartment A and compartment B. So compartment A contains gene-rich, actively transcribed, early replicating urochromatin, whereas compartment B tend to be gene-poor, transcriptionally repressed, and late replicating heterochromatin. So, and mutation rate is generally higher in compartment B. And I will provide you with an example of our study where we explore how this large-scale confounding factor relate to mutation rate in specific gene, particularly in histone genes. So we found that mutation rate histone genes had a negative correlation with gene expression level, as well as significant positive correlation with replication time. And we did not find any significant correlation with chromatin state, but it might be because of the limitation of the data which we used in our analysis. So moving forward to mutation rate at the mesoscale. So the canonical DNA is a right-handed double helical structure and known as bDNA. So although bDNA is the most predominant configuration in our cell, in the human cell, so there are more than 20 non-canonical structures that have been also reported. They include quadruplets, DNA hairpins, DNA loops, and others. And these non-canonical DNA structures, they can affect mutation rate. So, and it's due to their impact on DNA replication and repair processes. So this structure may pose challenges for DNA replication, DNA repair processes, and also they can cause replication forks to stall and interfere with in efficient DNA repair. So the variation in mutation rate also at the mesoscale also can be associated with 10 base pair periodicity in nucleosome. So this within nucleosome, the somatic rate is exhibit this 10 base pair periodicity. So, and this is because of DNA accessibility. So damage-induced mutagenesis is accelerating in position with the minor group facing out of histones, whereas replication mismatches are more frequent in positions with the minor group facing in. facing in histones. So, and now let's talk about the background mutation rate variation at the single nucleotide scale. So we know that the probability of mutation depends on the local DNA context. And previous studies have shown that this local DNA context is a major factor underlying the largest proportion of mutation rate variation at this small single nucleotide resolution. So here we can see the variation in mutation rate at the resolution of single nucleotide. So for driver mutation prediction, we need to take into account the mutation, this background mutation rate at single nucleotide resolution. In other words, we can compute mutation rate. This is background mutation rate, which are shown in gray bars. And we can also compute observed mutation rate. And driver mutations, so will be those which are observed significantly more often compared to expected mutation rate, which are here. This observed mutation rate shown here as this red lollipops. And this gray is expected mutation rate estimated on single nucleotide resolution. So now I showed you how it's important for driver mutation identification estimate the background mutation rate at the single nucleotide level. So now let's talk about approaches which we can use to estimate this mutation rate at this such small scale resolution. So, and I'd like to start with classification of nucleotide substitution considering the DNA sequence context. So there are 4 types of DNA nucleotides. They can form 6 types of mutations. And if we consider trinucleotide context, there are 4 nucleotides at 5' and 4 nucleotides at 3' sites. So multiplication of them gives us 96 context-dependent mutation categories. So, and then we can use these 96 context mutation categories to construct a mutational profile. So on x-axis, you can see this 96 mutational categories, and y-axis their probabilities. And for a group of samples, we can create a matrix where each row corresponds to the tumor samples, and each column represents this context-dependent mutation category. And the intensity of the pixel indicate the frequency of these specific mutation categories. So, and thus the mutational profile of a tumor sample is a result of mixture of context-dependent different mutational processes. And from those mutational profiles, we can infer codon mutability. So, and the codon mutability is calculated as a probability to observe a specific type of codon change. And so I'm not going to dive into details about this formula, how to compute codon mutability, but the main idea is that the amino acid substitution rate depends on the local nucleotide context. So another straightforward way to estimate background mutation rate at single nucleotide resolution is to go beyond trinucleotide limitation and use mutational motifs. So a mutational motif represents a mutation and its local DNA context, where number of nucleotides before and after the mutation can be varied. So this approach allows us to take into account sequence context of any length, and it has a simple representation. And we can scan genome for some specific motifs and calculate their enrichment. So, and we have recently shown the success of this approach in prediction of driver mutation for DNA polymerases epsilon. So, and this novel driver mutation, they have been identified and experimentally validated. So let's move forward, and the third group of methods which take into account this background mutation rate. This group of methods, probabilistic and machine learning methods. So there are machine learning methods that specifically aim at predicting cancer driver mutation. And we know that machine learning involves creating algorithms to make prediction, learn from data and make prediction without explicitly being programmed. So in these methods, first they select, so the various features are selected based on the cancer data we use. Next is we select a suitable algorithm to train the prediction model based on the features of interest and the our cancer data. And then after the training process, the algorithm can make prediction of driver mutations based on new data. And finally, these results are validated and interpreted to elucidate the driver status of mutations. So however, these methods, they have some drawbacks. So they require a large and unbiased set of training data, and the number of gold standard data set for training and validation is limited. There are methods which doesn't rely on training. And for example, this probabilistic statistical framework, which has been recently published, so it uses somatic cancer mutation data, and then it takes into account the number of patient cohort and frequency of mutation. And as we discussed the mutation, background mutation rate, this framework builds cancer-specific DNA context-dependent mutational profiles and uses these profiles as a background mutation rate model. So then the algorithm calculates the probability of observing a specific type of mutation more frequently at specific site more frequently than the actual number of mutation in given cohort of patients. But this method is simple and doesn't rely on training. But however, the main difficulty is to identify driver mutations that occur at low frequency. They might escape from detection. So this identification of driver mutations at low frequency is a very challenging task. And I'd like to introduce an approach that can help to address this challenge. And let's begin with a guilt by association. So this guilt by association concept. So this concept refers to when someone concludes that a person is guilty or guilty of a crime of wrongdoing just by simply being associated with individuals who are known to be guilty. So for example, if a person A is a friend with person B, who has a criminal record, according to this concept, person A is also guilty of a crime only because of their association with this person B. So, and applying this guilt by association concept to mutation in cancer, we hypothesize that if mutation occurs at specific position in one protein, can mutations at similar aligned protein sites produce similar effect in cancer? So, and let me explain in more details. So we know that the protein is made of amino acid sequences, and the amino acid sequence determines protein structure and in turn dictate function. And proteins sharing similar amino acid sequences often share similar function, and a concept is known as a homology arising from a common evolutionary origin. And to visually represent this relationship between sequence similarity and function similarity, let's take a look at this plot. So in this figure, we illustrate the results of function similarity with respect to sequence identity as identified by BLAST, this pairwise algorithm for pairwise sequence alignment for gene ontology molecular function. So let me explain these indexes. So in gene ontology, terms are hierarchically organized and each term associated with a specific index level. So in this figure, the probability of functional conservation increases with increasing sequence conservation. So, and the main idea, if two proteins have similar sequences, they are more likely to perform similar function. So, and coming back to our hypothesis, so if we have mutations which affect a particular site in one protein, can mutation at a similar aligned protein site produce similar effects in cancer? So, and to test our hypothesis, we can apply the following computational framework. So we can perform pairwise alignment of all human proteins and create similarity matrix. So this similarity matrix that you can see here, this all proteins against all proteins, and the color intensity indicate the similarity between two sequence alignments. And the more intense color, the higher the similarity. And here we can see some clusters of proteins with high similarity. And we can take these clusters, we can take this cluster of highly similar protein sequences, and we can obtain observed frequency of mutation at specific site I, which is mutated. And N indicates the number of similar proteins which are in this similar group. So, and then we can take into account background mutability, which is represented by codon mutability, and compute some score. So for this specific site A. And so this score is calculated based on taking a natural logarithm of ratio observed frequency and codon mutability, and then this value is adjusted by using some specific weights. And then we sum this across all proteins at the specific mutated site A. So currently we are working on this approach, and we are applying this approach to histone proteins. And in each histone protein family, many genes encode identically or highly similar histone proteins that can be used interchangeably in the nucleosome. And in most cases, they are thought to perform the similar function, similar or the same function. Here you can see an alignment of gene products of histone protein core histone protein H2B. The intensity of a color in each square represent the number of mutation at this specific position within each protein. And these numbers represents the total number of mutation at specific site across all proteins. And now we are working on identification whether this site is under positive selection. Because if you look at this, we can see that taking into account only one protein might not be enough. So we have to sum this mutation across all homologous proteins. So like in case of histone proteins. And it's actually a very challenging task because we have to deal with many pairwise alignment, many sequences of many human protein sequences. And it's pairwise alignment of all human sequences gives us a large number of pairwise alignment which you have to deal with. And another difficulty is to choose right mathematical model which can allow us to correctly identify this driver mutation across a group of similar proteins. So, and another challenge is to choose a right gene weighting model. So to assign weight for proteins. So, and I'd like to conclude my talk with a short summary. So thousands of mutation tumor genomes, but only a few of them drive cancer progression. So driver mutation influence the clinical phenotype in impacting patient outcome. Mutation rate exhibit significant variation across the genome. Driver identification is a challenging task. and requires precise estimation of background mutation rate and single nucleotide resolution. And new approaches are needed to detect low-frequency driver mutations. So, and I'd like to acknowledge my supervisor, Professor Anna Panchenko, our lab members, our collaborators, and committee members. So thank you for your attention. And I would...
So, questions? And those that are listening from on the Zoom, if you have a question, you can either raise hand or just chime in. Go ahead. Easy, go ahead.
Thanks for your talk, Daria. I think I'm very new to this field and I don't know much about it. So I know some of the challenges with using the rate gene editing method. Can you tell us about like what different methods you're thinking of and like maybe which method you're thinking of going with?
Yeah, there are many different methods which allows us to weight genes, and some of these methods can be number of mutations per gene transcript. So it's one of the methods. However, there are other methods. If we think about large-scale variation, we know that some genes which are early replicating or late replicating, we can assign them with different weights for our prediction identification. Yeah.
So did you have a follow-up there? So histones are probably advantageous to study for a few reasons.
Mm-hmm.
Maybe you can tell us a little bit about why it would be more advantageous to do what you're talking about with histones than, say, I don't know, DNA polymerase.
Yeah, DNA polymerase was fascinating, yes, and what we have found is interesting. And then in the case of histones, so it's been known that mutation in histones, they can lead to cancer progression, so and cancer development. So and they are thought to be understudied for in some research. So that's many reasons to study histones. So, yeah.
But I'm guessing that being able to line up not just the 16 or so for H2B,
Yes.
the other, you've got a lot of sequences that you can do alignments and look for, say, highly homologous,
Right,
or highly
yes.
conserved sequences that are more likely to be sites where a change of amino acid is going to be deleterious to function.
Mm-hmm.
But there, I guess, not a lot of classes of gene or a lot of classes of genes that would be of interest wouldn't fall into the same category of having, I don't know how many histone genes there are, 100, something like that.
Histone genes, I think around 90 something,
Yeah.
90 histone genes, yeah.
So you mentioned BRAF, for example. So there's, I can't remember how many.
Yeah, there are
RAFs
other genes, yeah, and
well, there's a small number of RAFs compared to histones. So
okay.
do people find that some of these tools are going to be, you're going to kind of, histones would be a magnet for using some of these approaches, but you're going to have to use something else in another class of genes.
Yeah, but here, when I was talking about histones, so this is just our case study. We are going to apply our computational framework to test how it works for histone genes, because they are very representative in that case.
Mm-hmm.
As one histone protein can be encoded by multiple genes, yes. So, and yeah, currently we are working on all human proteins. All human proteins, so it's a...
Got you covered, yeah.
Maybe.
Yes.
But just following up on the histone question. So obviously there are histone variants. So there's within a specific class there are slight variants, but there are also large scale variants. That have like macro H2A, things like that. How far do you go in terms of including those in the alignment?
Okay. Well, so I think we can further narrow down our classification. So in terms of in terms of prediction drive mutation, that in terms of that applying that model which are developing on, and like classify these histones based on variants or based on their other isoforms.
But do you apply like an evolutionary aspect to it in terms of saying, okay, this one is, evolutionarily similar enough to be included in that same group, or do you?
No, we are just rely on sequence similarity in this case. So we are not assuming evolution of this, evolution of histones.
But obviously those are going to be related.
Yes, yeah, definitely, yeah, definitely.
Less sequence involved.
I should say that Daria is developing a method, and there is a threshold which user can specify by the user, threshold on the level of sequence similarity, on a fractional line, so the user can use this method and actually determine the threshold by themselves.
Right, but it's not about us applying it, right? It's about something computationally arising from that. But if you look at that, at what point does it become too far to improve your predictive model?
Right, but it basically depends on our knowledge. So which threshold we want to apply to this specific to our specific biological model, right? You can define threshold, let's say, is 80% of similarity. So we are taking those proteins which have 80% of sequence similarity, and then test our
What I'm saying is that you can't do it empirically. You can't say, okay, I'll try 80%. It has to come from the actual analysis. It has to say, okay, if I compare all the proteins and look at all those classes, at what point does that model diverge?
Yeah, so this is the person who applies this method decide based on the knowledge of this family or the evolutionary relationship of this family. So this method just tells you how to accumulate the observed frequency, how to normalize the background probability. But the threshold could be, for example, the site could be under structural, there could be structurally concept site. And then you go back into evolution to maybe percent identity of 20 or 15. But you see that structurally all these domains are conserved, and the selection might actually impact this particular site because it's structurally important. Or like if you want to talk about function specificity in your family, you think that should be grouped based on very precise function specificity, you don't want to go beyond maybe 80 or 90% sequence. And then you group this counts, basically grouping counts of observed mutations in camps. Based on your knowledge of your whatever your family or those who knows histones, they can use no concept threshold.
But you want a process that's knowledge agnostic, right? You don't want to have to apply your knowledge of histone biology. You want something that's going to work on any protein.
Right.
Well, no, we cannot apply the threshold depends on the protein. So we just say the user, we give the program and the user can apply the threshold based on the knowledge of this particular family. But we can talk later.
Maria, go ahead.
Yeah, it's two comments related to that, and I think what Anna was saying makes sense. This idea that the reality is that the threshold will depend on the type of protein. And I was curious if you were going to, in your analysis at all, consider whether the part of the protein is structured domain or not.
We might do it, but right now, no, we're just performing without any assuming any location in this specific sequences. We're just performing this pairwise alignment and no other information we are including so far.
Go ahead, Chris.
So in terms of the like replication timing, expression, and all of those things that you use to establish mutational rates, they're all tissue specific, right? So a gene is going to be late replicating in one tissue, but it's going to be early replicating in the other, depending on its status. So how do you encompass all of that data together in that sense? And maybe you could also comment on the slide. I don't want to go back to it, but where your combination of A and B chromatin domains wasn't significant. I can't remember which. The first
Yeah, yeah, I understand that. So this one and
Right, yeah, so you
Yeah,
combine
yeah.
A and B, so that to me is really, those compartments are the expression of late replication timing and expression, so why is that not significant where the other two?
yes, it was not significant in our case of when we were exploring the mutation rate variation for histone genes, and it was not significant as I mentioned because of the limitation in data which we use. This high C, this chromatin state data was obtained from just the single cell lines, which might not represent the overall picture of mutation rate for different cancer types.
Yeah, okay, so back to my question then about integrating different tissue-specific aspects of this. How are you going to integrate all of those? Because in a way, it's a bit contradictory, right?
Right, yes. So first of all, this we are talking about the prediction of drive mutation at single nucleotide resolution. And these large scale factors, they affect at the megabase scale. So,
Right.
And yeah, that's true that this expression level replication time in chromatin state, they're all cell type specific. So they vary from cancer type to cancer type. So in this analysis, this expression level and it's an average from different cancer types, represented an average of different cancer types. So this is a limitation of the specific study.
But isn't that going to neutralize some of your effects because it's going to say, it's an average. So that's not a very sophisticated way of analyzing that. Is there no way of incorporating that aspect of tissue specificity? Because otherwise you're losing that richness of information.
Yeah, right. But here is represented just as an average. We can take into account every expression level for every cancer type. So not averaging them, that just we have specific type and that apply framework specific to specific cancer type based on data we use for cancer type. So so not averaging to predict to develop a model for the all cancer types without averaging.
Right. So it's a question of how specific your model is going to be. I mean, seems like you're trying to generate too general a model. Because if you're trying to come up with a mutational rate that's applicable across the board, that's not going to work because you've averaged all of these tissue-specific things. So are you not creating, is it not more important to create a model that's specific to a given cancer type rather than a generalizable model? I understand how those two are going to be rationalized.
Yeah, but I wouldn't say that we are going to create a generalized model. As I mentioned, we can create a specific model for a specific which can be related to a specific cancer type. So based on the data availability.
But you then have to use mutational your other data that's also specific to that cancer or that tissue type.
Right. Yes, the mutation rate variation at single nucleotide level we can calculate based on also we can estimate it based on cancer type specifically as well. Yes, yes.
Right. And is your model going to be sophisticated enough to be able to do that then?
Right, yes, I mentioned a few approaches which can be used for estimating background mutation rate at this nucleotide resolution, and it will be because we are used this data. The data is obtained from cancer type, specific cancer type. So,
right.
And we then apply these methods for estimation background rate, take into account those cancer mutation data.
Right. So I can't. So Charlotte, it's glioma. Your histone model.
No, histone model. No, it's not glioma.
The histone mutations are specifically glioma.
They are not only happens there.
Right.
Yes, yes.
But are you thinking about modeling a specific cancer that has more richness in terms of the data set? Because if you look at all the data sets you talked about, glioma is very small in most all those. You're going to be talking about breast, lung, prostate cancer. Are you thinking about modeling those particularly, or have you thought about where you start modeling this? getting back to the tissue-specific aspect.
Right, so we can, yes, for sure, we can start with a more generalized model with where we use all cancer types, and then we can stratify it based on the cancer type,
But I
based on the
suggest that
cancer type.
starting with all of them is not a good approach because of that averaging.
Yes, but for some cancer types, we don't have enough data to make any.
There's one which has had, it's very rich.
Yeah.
Love to have another conversation.
I don't see any questions on the Zoom. Oh, go ahead, Jessica.
Thanks, Daria. That was really interesting presentation. So one of the questions was kind of simple. So when you predict if a mutation is going to be a driver mutation, do you generally see that there's more driver mutations in the tumor suppressors or promoters? And if I was going to ask if it was cell specific, but I think it's average.
Could you please repeat your first question?
Do you see more tumor suppressor more driver mutations in tumor suppressors or in like tumor promoters?
We do not classify based on this tumor, whether it's tumor suppression or tumor suppressor or another type of yes. So we didn't performance.
Anything else? I don't see anything left on the Zoom then. Then join me in thanking Daria for a very stimulating talk. And to the other students, don't be afraid to leave lots of time at the end of your talk